Now, let's talk about mechanism of skeletal muscle contraction. First of all, what is a motor unit? Let me explain this. For example, this is anterior greyhorn. Okay. From anterior greyhorn, a motor neuron comes out. When this motor unit, when this motor nerve enters to muscle fascicle, it gives a lot of branches. And each branch of a motor neuron innervates a muscle fiber. So for each muscle fiber, we have one branch of a motor neuron. So as a collectively, all muscle fibers which are innervated by one motor neuron, as a collectively, they are called motor unit. As a collectively, they are called motor unit. Now, for example, this is one muscle fiber, and this is a branch of a motor neuron. And from previous lectures and previous lecture, we have studied two important topics. First, we've studied about the basic structure of nervous tissue. Then we studied about the resting membrane potential and action potential. Before watch this video, please watch the video of resting membrane potential and action potential. Now, let's move on. From cerebral cortex, from motor cortex, now you decided to move your right hand, okay, to contract this bicep brachii muscle. From cerebral cortex, from motor area, depolarize and repolarize, come to the spinal cord. And from spinal cord, these depolarization and repolarization stimulate these neurons which are present in anterior greyhorn. Now, a neuron of anterior greyhorn is stimulated and depolarize and repolarize reach to the bulb of this neuron. Okay, now when these depolarization and repolarization reach to the bulb of a neuron, okay, in bulb of a neuron, this depolarization and repolarization cause a stimulation of calcium voltage gated channels. And you know that we have more calcium outside of the cell than the inside of the cell. So calcium ions enter from high concentration gradient to low concentration gradient from outside to the inside of the bulb. Now, in this bulb, we have some vesicles, and we have a lot of neurotransmitters inside of these vesicles, which are called acetylcholine. Okay, so acetylcholine is previously stored in these vesicles. Now, when calcium enters, it causes activation of an enzyme or protein which is called calcium calmodulin dependent kinase. And you know, kinase are those enzymes which phosphorylate other enzymes or activate other enzymes and other proteins. Now, this calcium calmodulin dependent kinase adds an ion of calcium, adds a phosphate on a protein which is called synapsin. And function of synapsin is that synapsin holds this vesicle to the cytoskeleton of the bulb. Now, when you add phosphate on this synapsin protein releases this vesicle, and now this vesicle comes to an area which is called dense bar area or active zone. Now on this vesicle, we have some proteins which are called V snares, and also we have some proteins on the membrane of the bulb of a neuron which are called T snares. Now, this is a V snare which is called synaptobravin. This V snare is located on this vesicle. These two snares, this red and blue, these are called syntaxin and SNAP25. Now, when this vesicle is released by this calcium calmodulin dependent kinase, and when it comes to this area, this protein, which are called V-snare, automatically binds to two T-snare proteins, which are called syntaxin and SNAP25. So this binding is automatically. But this calcium also activates another V-snare protein, which is called synaptotagmin. And now synaptotagmin furthermore stabilizes these connections between these V-snares and T-snares. And due to these connections, they go to a transformational change and causes to pull this vesicle from active zone to near to this membrane of the bulb. And now this vesicle will be attached to the membrane of the bulb of a neuron, and this vesicle releases its own neurotransmitters, which are called acetylcholine. Okay, now this area between, as a collectively, this area between the bulb of a neuron and between the sarcolemma, as a collectively, this area is called neuromuscular junction. It means junction between cell membrane of a skeletal muscle and between the end part of a neuron. Now acetylcholine is released by the help of depolarization and repolarization from the bulb to this area, and this narrow space is called synaptic cleft that we've studied about it in previous lectures. Now this acetylcholine, okay, this acetylcholine or two acetylcholines bind to a protein which is, or to a receptor which is located on the cell membrane of a skeletal muscle or on the cell membrane of sarcolemma. Now this red receptor, this is called nicotinic receptor, or these are called cholinergic receptor because these receptors are stimulated by acetylcholine. Now, and here you know that acetylcholine causes contraction of skeletal muscles and activation of a lot of skeletal muscles in our body. So that's why some people who use nicotine, after a little bit time, they become alert. Okay, that's because nicotine also has this capability to stimulate these cholinergic receptors. And we know you can find nicotine in cigarettes, or you can find it in tea, in coffee, and so on. Now, this nicotinic channel here, it is actually for sodium, but potassium and also calcium have this capability to pass through these channels, but chance for these ions are less than the sodium. Now, when acetylcholine stimulates this nicotinic channel, the gate of this nicotinic channel will be opened by this acetylcholine, and you know that we have more concentration gradient of sodium outside of the cell than the inside of the cell. So now, by the help of acetylcholine, a lot of sodiums or positive charges flow from the outside of the cell to the inside of the cell. Now this area, this membrane of the skeletal muscle is at rest as before stimulation. But when you enter a lot of sodium or positive charges to the inside of this muscle fiber, so it causes production of action potential. How? Like this, that during rest, the resting membrane potential in skeletal muscle fiber is around negative 90 millivolt, that we've studied it under the title of resting membrane potential and action potential in previous lectures. When you enter a lot of sodiums, it causes the resting membrane potential, it causes the inside of the cell to lose its negativity because you added a lot of positive charges to this cell. So I'll go from resting membrane potential to a state which is called threshold potential. And threshold potential here is around negative 65 millivolts. And we know that threshold potential is a special voltage in which a lot of voltage gated ion channels will be opened. Got it? Now, when you reach to threshold potential around negative 65 millivolts, okay, furthermore, another type of sodium voltage gated channels will be opened. And you know, we have a lot of sodium outside of the cell than the inside of the cell, and more sodium flow occurs inside to the cell. Now, the cell becomes more positive. And we know from previous lecture that this is called depolarization. And at the end of the depolarization, furthermore, potassium voltage gated channels will be opened and cell membrane comes once again to repolarization state or to its negative state due to potassium efflux, because potassium voltage gated channels will be opened due to this depolarization. Now, this depolarization and repolarization continue deep to the T tubule or transverse tubule. Now, these positive charges or negative charges or depolarization and repolarization cause to activate a special receptor or protein which is called dihydroferritin receptor. And we have some drugs related to this receptor that we will talk about it at the end of this video. Now, dihydroferritin receptor is activated by this potassium, by this depolarization and repolarization. Now, it causes this depolarization and repolarization. It causes activation of another protein which is located on sarcoplasmic reticulum. And we have two sides of sarcoplasmic reticulum in a skeletal muscle. These parts which are near to the T tubule, these parts are called a cisterna or terminal cisternae. Now, a protein which is located on terminal cisternae, these proteins are called ryanidine receptor. So, first, by the help of depolarization and repolarization, you activated dihydroferritin receptor, and dihydroferritin receptor activates ryanidine receptor. Now, you know that we have a source of calcium inside of sarcoplasmic reticulum that is stored previously. Now, when you open this channel or when you stimulate this ryanidine receptor, a lot of calciums from high concentration gradient to low concentration gradient flow from the end, flow from the sarcoplasmic reticulum to the cytosol of the skeletal muscle. And now this is calcium, okay? And this calcium is released by the help of depolarization and repolarization and activation of these proteins. So now in this step, we have more calcium in cytosol of this cell membrane. Now, these calciums bind to troponin. To which part of troponin? To troponin C part. Now, when a lot of calcium binds to the troponin C part, to the troponin C part of the troponin protein, it causes some transformational changes in this troponin protein, and due to this change, these tropomycins go deep to this groove between these two thin filament. And now this tropomycin will be disappeared from this area by the help of troponin. And also the active sites of globular actin which were blocked by troponin I, they also will be opened by the help of calcium. Now these active sites are available for the head of myosin. Now in this step, automatically the head of myosin binds to the active site of actin. So when a lot of head of myosin bind to the active site of thin filament or actin filament, now they use ADP, that I'll talk about it later. They use ADP, and by the help of ADP, they will bend a little bit, okay? When the head of thick filament or myosin proteins bend because of sliding of these two thin filament over each other. For example, these are head of myosin. They will bind in thin filament and they will tilt to a little bit forward, which is called power stroke. And due to this power stroke, these two filaments come toward each other. Now, when a lot of thin filaments overlap on each other or cross each other, they cause these Z-discs to close to each other. And now, when you bring these Z line or Z discs close to each other, it means you shrinked this myofibril. And when you shrinked myofibril, it means you shrinked a lot of muscle fibers in the same time, because in the same time, thousands and hundreds of motor neurons fire on a bundle of a skeletal muscle. So all of them shrink and all of them contract. After this, I'll tell this word contraction, okay? So all of them contract, and when a muscle bundle contracts, so it causes to pull a bone and it causes movement of this bone. That's why when you want to stimulate contraction of this biceps brachii muscle, so it will contract and it causes movement of bones. So this was the basic mechanism of muscle contraction. Now, someone will ask you that after contraction, what changes will be appear in these sarcomeres? Okay, now listen to me carefully. When two Z discs come close together, first of all, this area, the space between two thick filament, which is called I bend. This I bend, for example, this is an I bend, this is an I bend. So as a collectively, I bend get closer and or they get smaller. Got it? This is one change during contraction. Next, this small space between two thin filament, which is called H zone. Due to overlapping of this thin filament, this H zone will be disappeared completely. Okay, so these are changes, and also space or the size of sarcomere gets smaller. Got it? So these were changes during contraction of a skeletal muscle fiber, and at the end of contraction, these calciums enter back to the sarcoplasmic reticulum by the help of a protein which is called SERCA. This is a pump, okay? It uses ATP or energy, sarcoplasmic endoreticulum calcium, ATP's pump. It pumps two calcium to the inside of the sarcoplasmic reticulum and two proton ions from the inside to the outside. And also, when calcium enters to the sarcoplasmic reticulum, calcium or around 40 to 30 to 40 calcium ions immediately bind to a protein which is called calciquestrine. Got it? Now, we left in one thing in muscle contraction, and this is the cycle of power stroke. Now we will talk about the basic cycle of a power stroke that have this head of myosin causes actin sliding, and also how head of myosin uses ATP or energy. Got it? Now, let me explain it here. First of all, this my head is active side of actin filament. This part of my head is head of myosin. Now, before contraction, head of myosin is separated from the active side of actin by the help of this protein which is called tropomyosin. Now, when calcium came here, it caused these tropomycins to go to a change. Okay, now this tropomycin go away from this area. Okay, now I left only with head of myosin and active side of actin. When you remove tropomyosin, head of myosin automatically joins to the active side of actin protein. Okay, and rest. It will join automatically. But before contraction, okay, head of myosin has a DB plus phosphate. Now, when it joins, okay, it was located in spring-like structure previously. Okay, now when it joins automatically, so now it will create a power stroke. It will bend a little bit to anterior for example, it will bend like this. So when it bends, it causes sliding of this actin. And during this bending is called power stroke. Okay, power stroke. Now, during power stroke, this head of myosin releases its phosphate first, then it releases ADP. Now, you know that power stroke occurred and thin filament came from this side to this side. Now, ATP will bind to this area. When ADP binds to this area, this binding of ATP automatically detaches the head of myosin from thin filament. Okay, and now ATP will be hydrolyzed to ADP plus phosphate, and when ATP is hydrolyzed to ADP plus phosphate, okay, now the head of myosin cocked back, okay, and it's ready to next cycle. And in next cycle, now look it, in next cycle, when you remove tropomyosin from this area, it will bind automatically. We have ADP plus phosphate. It causes power stroke, and during power stroke, ADP and phosphate will be released, and once again ATP will bind, and ATP binding of ATP detaches this head of myosin from active side of actin, and when ADP is hydrolyzed to ADP plus phosphate, once again it cocked back and it's ready to next cycle. So like this, as a collectively, they cause sliding of actin filament on each other and causes contraction of skeletal muscles. Got it? Now let's talk about mechanic of skeletal muscles.